The death of neurons, cardiomyocytes, and pancreatic beta cells results in the clinical symptoms of the disease. The loss of neurons in the brain leads to ataxia, defined as a lack of voluntary muscle control and coordination. Patients experience muscle weakness, difficulty walking, and loss of sensation and reflexes. The loss of pancreatic beta cells leads to decreased insulin production and the development of diabetes. And finally, the loss of cardiomyocytes in the heart leads to hypertrophic cardiomyopathy, or the abnormal thickening of the heart, which can lead to heart failure, and this is the most common death for patients with Friedreich's ataxia.
Hello there, welcome back to another episode of Rare Diseases. Rare Disease Review is a not-for-profit medical journal organized by students from the University of Toronto, McMaster and Western University. This podcast is created by the branch of Western University. The podcast aims to advocate for rare disease awareness and promote students' involvement in the rare disease community. We appreciate your listening and support today. For further information on scientific translation of rare disease information, please visit www.rarediseasereview.ca. The content of this podcast is not intended to be a substitute for professional medical advice, diagnosis, or treatment, so please always seek the advice of a physician or other qualified health provider with any questions you may have regarding a medical condition. Never disregard professional medical advice or delay in seeking it because of something you have heard from this podcast. My name is Song, and you're listening to the Rare Disease Podcast. For this episode, we're going to dive into a rare hereditary disease called Friedreich's ataxia. Friedreich's ataxia is a genetic progressive neurodegenerative movement disorder. The typical age of onset is between 10 and 15 years. The initial symptoms of Friedreich's ataxia includes unsteady posture, frequent falling, and progressive difficulty in walking due to impaired ability to coordinate voluntary movement, which is why it is called ataxia. The affected individual often develops slurred speech called dysarthria, characteristic through deformities, and an irregular curvature of the spine called scoliosis. Friedreich's ataxia is often associated with cardiomyopathy, a disease of cardiac muscle that may lead to heart failure or irregularities in heart rhythm called cardioarrhythmia. People with Friedreich's ataxia have a higher chance to get diabetes. Friedreich's ataxia presents degenerative changes in sensory nerve fibers at the point where they enter the spinal cord in the structure known as dorsal root ganglia, which results in secondary degeneration of nerve fibers in the spinal cord, leading to a deficiency of sensory signal to the cerebellum, a part of the brain that helps to coordinate voluntary movements. Friedreich's ataxia is caused by a trinucleotide repeat expansion in the FXN gene, which codes for a protein called fractaxin. To better understand the scientific research on Friedreich's ataxia, I spoke with Dr. No, a Ph.D. graduate from Department of Pathology and Laboratory Medicine at Western University. Dr. No focused her studies on the cellular and molecular mechanism of oxidative stress, which in Friedreich ataxia is an important contributing factor in the progression of the disease. Welcome, Dr. No. It's our honor to have you with us here to speak about Friedreich's ataxia today.
Thank you so much for having me.
Yes, to start off, can we get to know a bit about your work?
Well, I recently completed my Ph.D. in Pathology and Laboratory Medicine at Western University, and my research focused on a protein called Nrf2, the transcriptional master regulator of the cellular oxidative stress response. So oxidative stress is defined as an imbalance between the production and accumulation of harmful free radicals in cells and tissues, and the ability of a biological system to detoxify these free radicals. Oxidative stress is implicated in a multitude of human diseases, including Friedreich's ataxia, where it plays a major role in the progression of the disease.
Wow, that's very fascinating. I'd really love to hear more about your research when you get a chance in the future. Now for today, could you give us a little bit of the insight into the cause of Friedreich ataxia?
Friedreich's ataxia is caused by a mutation in the FXN gene on chromosome 9 that encodes for a protein called fractaxin. It is an autosomal recessive disease, meaning an individual must inherit two abnormal copies of the FXN gene from both parents to express symptoms of Friedreich's ataxia. This inherited mutation causes an abnormal repetition of three DNA nucleotides, GAA, or guanine adenine adenine, known as a trinucleotide repeat expansion. In normal individuals, this GAA sequence is repeated approximately 7 to 38 times within the FXN gene. However, in individuals with Friedreich's ataxia, this GAA sequence is repeated anywhere from 66 to over 1,700 times. This trinucleotide repeat expansion causes gene silencing in the FXN gene, resulting in little or no production of this frataxin protein.
That's really interesting. So frataxin protein is being affected in this disease. For the patient to develop Friedreich ataxia, what is the role of fractaxin?
Frataxin is involved in the incorporation of iron into iron-sulfur clusters, which serves as various enzyme cofactors, including mitochondrial enzymes involved in electron transfer and ultimately mitochondria ATP production. With insufficient frataxin, two key outcomes occur. Firstly, mitochondria are unable to incorporate iron into these iron-sulfur clusters, resulting in decreased mitochondrial ATP production and insufficient energy for the cell. Secondly, these unused iron accumulates in the cell and reacts with oxygen to form harmful free radicals, resulting in oxidative stress over time and ultimately oxidative damage if not remedied. This reduction in cellular ATP combined with oxidative damage results in cell death, specifically in neurons and cells of the pancreas and heart, where frataxin is normally most highly expressed.
Thank you so much. So with these changes at cellular level, what kind of symptoms and signs a patient might present?
The death of neurons, cardiomyocytes, and pancreatic beta cells results in the clinical symptoms of the disease. The loss of neurons in the brain leads to ataxia, defined as a lack of voluntary muscle control and coordination. Patients experience muscle weakness, difficulty walking, and loss of sensation and reflexes. The loss of pancreatic beta cells leads to decreased insulin production and the development of diabetes. And finally, the loss of cardiomyocytes in the heart leads to hypertrophic cardiomyopathy, or the abnormal thickening of the heart, which can lead to heart failure. And this is the most common death for patients with Friedreich's ataxia.
Wow, this is very critical conditions. So how may a patient be diagnosed and treated currently?
Friedreich's ataxia is diagnosed by genetic testing to identify the trinucleotide repeat expansion in both copies of the FXN gene, in addition to brain images scans, nerve conduction studies, blood tests, and cardiac tests. Unfortunately, at the time, there is no cure for Friedreich's ataxia and currently no approved therapies for the treatment of this disease. Treatment is focused on minimizing symptoms and maintaining comfort and muscle function. Options for this include physical bracing of limbs, physiotherapy, speech therapy, occupational therapy, corrective skeletal muscle surgery, and medications to treat heart disease and diabetes. Importantly, ongoing research is being conducted with hopes of a cure.
So with all the wonderful information you presented here, could you tell us about the research side of Friedreich ataxia and how this is related to your research?
As mentioned, Friedreich's ataxia is a result of decreased production of the protein frataxin, which can impair mitochondrial function and cause oxidative damage. This oxidative stress-induced damage in Friedreich's ataxia is of particular interest to me, and so I'll take a moment to discuss some of the current work that is being done in this area. The free radicals, such as those produced in Friedreich's ataxia, can damage cellular DNA, proteins, and lipids, resulting in cell death. The cell contains various mechanisms to detoxify these free radicals, and this is chiefly controlled by the protein transcription factor Nrf2, which I mentioned before. Nrf2 is responsible for activating the cellular antioxidant response, so essentially more Nrf2 means less oxidative stress. Thus, molecules that can activate Nrf2 may have the potential to induce clinical benefits in patients with Friedreich's ataxia. A 2019 study by Petrillo and colleagues compared different Nrf2 activators for the treatment of Friedreich's ataxia. They found that sulforaphane and acetylcysteine were particularly effective on genes involved in preventing inflammation and maintaining homeostasis for the antioxidant glutathione, and that dimethyl fumarate, omaxabolone, EP3 were effective in counteracting toxic products accumulation. And finally, that they found positive effects of idebenone on mitochondrial protection. Other studies have found similar benefits for Nrf2 activators in improving mitochondrial function, reducing oxidative stress and inflammation, and ultimately preventing cell death in the brain, heart, and pancreas.
Wow, that's incredibly fascinating. With these great progress in research, what do you think is the direction of future research?
Collectively, this suggests that the development of synergistic therapies based on a combination of Nrf2 activators may have therapeutic benefits to patients with Friedreich's ataxia. Such research is currently ongoing, and in addition to reducing oxidative stress and improving mitochondrial function, other areas of Friedreich's ataxia treatment research include modulation of frataxin-controlled metabolic pathways, frataxin replacement, stabilizers, and enhancers, methods to increase FXN gene expression, and gene replacement and gene editing therapies, as outlined by the Friedreich's Ataxia Research Alliance, FARA.
That's great. Thank you so much for joining us today, Dr. No, and thank you so much for the informative interview. I'm grateful for your time and the amazing research you have shared. You have provided a great contribution to advancing the science of pathology and medicine, and we're really honored to learn from you today.
Thank you so much.